Okay, so good morning, everyone. So a few announcements. The first thing is, as you know, the rough draft of the manuscript is due today. So once I receive it, I'm going to look it over and then give my comments and then meet with the teams to see how we transition from there to the first complete draft. And so a reminder that the complete draft would have to be in the actual template from the journal itself. The next thing is that next week we have a guest speaker, Dr. Peter Caravan, coming from Harvard Medical School, and he's going to present not during lecture time, but he's actually going to present during the RISE seminar series. And so I will make sure that you will be able to go attend. And also, I'm not sure if that's currently being delivered in a hybrid modality, but for those that are not able to participate in person, I will make sure that that will be available also in a hybrid modality. Okay, so we're going to continue our discussion. From here, we're transitioning into a little bit of a overview of formulations, in this context, nanoparticle formulation of compounds. And so much of what we've discussed throughout the semester, at least in the context of how I've delivered it, has been based on the use of small molecules for therapeutic applications or to influence some physiological pathway. And so there are limits, and one of the ways that people try to go around some of the limits in terms of the therapeutic potential of small molecules is by the use of nanoparticle formulation. And then the context here for the purposes of delivery. So when we look at most drugs, what we do identify is there's that a majority of what's available in the market may not necessarily be specific. Let's see. All right, Carmen, you can email me that later. Thank you. So what we're looking at here is the context is how most drugs have their limits because of a lack of specificity. And so we've looked at, in the context of anti-cancer applications, what are potential side effects due to this lack of specificity. And so common pharmaceuticals have a relatively narrow therapeutic window. And so what we're talking about in therapeutic window is really that difference between how you can concentrate into the area of interest, where you want the drug to actually have an application, versus having a systemic effect throughout the body. And so when we're, again, talking about chemotherapeutics, we have seen a number of studies that assess damage that can occur in healthy cells, organs, and tissues that can lead to toxicity and also to a poor quality of life and nutrition. And so nanomedicine has really taken a big interest in the field in terms of how you can potentially take advantage of certain features within the body that nano delivery can really help to improve the potential therapeutic application. So we're talking about nanometer-sized engineered particles that are designed for the purposes of enhancing the specificity of drugs while trying to minimize their side effects. And so I will say, as a disclaimer, there are still very much lots of limitations with the use of nanoparticles, but we're going to see what people have tried to do to improve their potential in terms of delivering small molecules. So nanomaterials can take on a variety of dimensions, shapes, and properties, and we'll see a little bit about this in terms of how that can influence effective delivery. They can be taken into cells by endocytotic mechanisms, depending, and this is particularly true in terms of how you engineer them, depending on how you surface coat them, how you decorate them with specific biomolecules. And one big feature that people like to claim is the capacity to deliver a high payload of a drug. So what does that mean? A high payload is to try to bombard as much concentration of the drug as possible in the site in which it's going to land. And so, in theory, into a specific site within the body. So the idea is that nanoparticles can also be engineered for the purpose of possibly detecting diseases. And so we're going to see a little bit of this in our next lecture, but also in the subsequent seminar presentation that we're going to see. So they take advantage of the capacity to have a high payload of a species, as I just mentioned to you, while exploiting certain specific properties, whether it's magnetic, optical, or even radio emitting properties for the purposes of imaging. And then you have those nanoparticles that combine this therapy with diagnosis, the theranostic agents. And so those are typically developed, and you often mostly see them in the cancer field, but there are the areas where people have used theranostics. So the idea is that you deliver something that has the capacity to treat cancer while also giving you a diagnosis of its progression. What we're going to focus specifically on today is the actual particles themselves and what features are being taken advantage of. So here we have a visual to give us a perspective of the dimensions about small molecules, cells, and where nanoparticles really fall between here. So we're talking about cells that are in this range here, right? So we have that limit of what you're trying to get something inside of that. And then you have where typical small molecules fall into this region down here, right? So we're below nanoparticle or nanometer size. And so obviously we're looking at nanoparticles in this range of 1 nanometer to, let's say, nanometers. And so we're going to talk a little bit about why we want a certain size in terms of being able to effectively get into the potentially the circulatory system, but also to potentially avoid being captured and eliminated before having any effect in terms of delivery. All right, so the delivery of drugs in nanosized formulations offers a lot of advantages. At least that's what people like to harp when they're talking about the use of nanoparticles. And so in this context, we're talking about the delivery of small molecules for this purpose. Now, here you have to think a little bit about the route of delivery, as we discussed in terms of small molecules and in terms of how stable they are within a certain pH environment. Well, something similar is true for particles, but more in the context of their actual size. So if we're talking about an oral delivery of these type of materials, well, there you have a limit. So the size limit for particles to be able to cross the intestinal mucosal barrier of the gastrointestinal tract typically is the main restriction. And so particles that in the intestinal lumen within a range of 3 to 10 micrometers cannot migrate through the lymphatic system. Whereas particles that are larger than that, so we're talking about larger than 10 micrometers, they will now be able to be taken up into the gastrointestinal tract. So there you already have a limit. But again, 10 micrometers, if you go back to here, you're already starting to be in the range where, you know, the size of cells. So that's already a bit too big. So if you want to deliver something that's in the micro region, it's preferable that you do so in a non-oral route, just because of the limitations in terms of the uptake of those materials. Now, the bloodstream also poses a restriction, and particles must be significantly smaller than low micrometers. I have here as a limit 5 micrometers. And so the idea is that you have to be well aware of the properties of your particles, that they don't aggregate. So you can design something that's, to say, small, but you want to also make sure that it doesn't aggregate to a size that's well above micrometer amounts. And the reason for that is if you're going through the circulatory system, having particles that are going to be this size can cause embolism. And so that can have very detrimental effects. So here you're causing clots that can lead to death. So the reason for this is that the smallest capillaries in the body are 5 to 6 micrometers in diameter. So that gives you another perspective of size. All right. Another thing to think about is those factors that are going to lead to the clearance of these materials. And so here we get a little bit of perspective. And here we're also going to allude to the immune system and how we can participate in this clearance. So synthetic nanoparticles with the diameter of greater than 100 nanometers are susceptible to the phagocytic clearance by the reticular endothelial system, or RES, which is an essential component of the immune system located in different organs of the human body. So the mononuclear phagocyte system, MPS, is responsible for clearing and degrading foreign materials introduced into the blood. So as I mentioned to you early in the course, we talked about how the immune system can recognize something as foreign. And so this is a system that is in place that can possibly recognize nanoparticles, neutralize them in such a way that they will not have any actual effect. The lowest practical diameter must be greater than 10 nanometers because particles with a diameter less than that are excreted readily by the kidney unless there is some sort of biomolecular interaction with these nanoparticles. Let's say, as we've alluded to throughout the course, serum albumin, that protein, which is a blood in our blood, that can potentially interact with these nanoparticles and help to prolong their blood circulation. So here we have a bit of a range between 10 nanometers and 100 nanometers in terms of desirable size. But even then, here I provide a table. We're not going to really go through this too extensively, but what this table tells us, if you're trying to target a certain organ, what dimensions you should aim for in terms of the size of the particles that are being designed. So here you have the organ and then the recommended size and certain specifications. So I'm not going to read through this. The point is for you in your spare time to look at some of this information. And if you're interested, looking further at the references from where this information was gathered. But there are some restrictions in terms of the dimensions and how that can potentially help you to reach a particular organ. All right, so I've talked about this in the course, this idea of the enhanced permeation and retention effect, and how that, especially when we're talking about for anti-cancer applications, something that people try to take advantage of when they design certain drug applications. And so this is the idea that the vasculature near the site of tumors is quite leaky, and so they're quite permeable. And so when we discuss permeability and we talked about certain size limits about small molecules, well, that factor doesn't come too much into play when you're talking about the vasculature near tumor sites, just because you can have much bigger things being able to enter. So things that are the nanoparticle range are able to localize themselves close into the vicinity where you have solid tumors. All right, so let's see that. So you have this, and this is the perspective of this now more permeable area where you can have small molecules. So that's depicted in these, this sort of stars, small molecules, and you have these nanoparticles that can also enter into the vicinity where you have these solid tumors. All right, so a little bit more about the EPR effect. So EPR, enhanced permeation and retention. So the vasculature of the tissue surrounding tumor cells is very porous in comparison to healthy cells. And so the size of the gap junction between endothelial cells of a leaky tumor vasculature may vary from 100 to 800 nanometers. So still within that nanometer range, which is desirable, again, for the purposes of designing things that are of a nanoparticle size. So this range is due to the diversity of the tumor type and location, especially with respect to the bloodstream. Usually the vasculature enables particles less than about 400 nanometers to accumulate in the interstitial space in tumor tissue, or even in the cytoplasm of tumor cells after cellular uptake. Diffusion from the interstitial space back into the blood is difficult, which is aided by a poor lymphatic drainage. So what this is saying is that not only do you have the potential for these nanoparticles to permeate in the region where you have these cells, or these tumor cells, you also have a higher retention of those particles because of the poor drainage of them. So you're going to have a net higher concentration in time in which they're going to be in the vicinity of these tumors. So conventional small molecule drugs, so we're talking about less than 4 nanometers, and microparticles have a limited plasma half-life and do not take advantage of the EPR effect. All right, so limitations. So despite the existence of the EPR effect, and here's the biggest thing to take note of, only about 5% of an administered nanoparticle drug typically ends up in the tumor, okay, 5%. So the remainder distributes to other tissues or is eliminated from the body by various mechanisms. So you have to be able to think 5% and then how much payload of the drug is there to wonder if that's going to be effective enough of a delivery of a small molecule, or is just using a small molecule itself going to be as effective. So that's something to think about. The blood contains hundreds of proteins called opsonins, which can bind to nanoparticles, form a corona around the particle to serve as a signal to the MPS. The particle is a foreign substance and it must be discarded. So this goes back into this idea that particles, as you know, are foreign to the body. And so our immune system has a way to mark them so that they can be cleared and not be able to effectively reach the site of interest. So here is something that people can try to do to go around this sonization process, which is to try to coat nanoparticles with polymers. So in this case, with polyethylene glycol, or typically referred to as PEG, and those can vary in terms of size. And so the idea is that you would coat them, and that tries to block recognition by this process. The thing that you also have to worry about is not coating them so effectively that you won't be able to have an uptake of the particle into the cells. There's also another limitation where, and this is something I need to look at more closely. So this is from a paper in 2019, that there is some evidence for nanomaterial induced endothelial leakiness, or NAM-EL. And that is that there's the potential that nanopart or nanomaterials can cause for the vasculature not near the site of tumors to become more permeable. And that could lead to a metastatic process, to metastasis. So that's something that has been a concern in terms of how nanoparticles can potentially result in this detrimental effect. All right. So with that being said, there are a number of drugs that are formulated in nanoparticle manner. And so here's just a table that provides you some of that information that are FDA approved. So these are the commercial names and for what application they're used. And if you look through some of these, you can see that a number of them are anti-cancer compounds, small molecules that are classically used, and the different formulations such as in liposomes, or even as albumin-bound nanoparticles. And you have even a formulation of cisplan in a liposome, so lipoplan, that's utilized in this type of application. All right. So as I mentioned to you, one way that you can try to enhance the capacity of nanoparticles to reach a particular cell is not only the EPR effect, but also by coating the surface of nanoparticles with certain groups that are recognized by receptors. So this idea of a cell targeting group. So what you're basically trying to do is engineer it in which you can have small molecules that have a very high affinity for receptors. And so here, what I'm providing you is a table of some of these small molecules that people will classically try to bind to the surface of their nanoparticles. So when I say ligand here, the ligand is a ligand in terms of something that you can bind to the actual molecules or the nanoparticles, not ligand in the sense of something that binds to metals. And so here you have, as an example, the use of folic acid, because there is an overexpression of folate receptor. This particularly is true in the field of an anti-cancer application, where people try to take advantage of that overexpression in certain cancer cell lines. And then you have hyaluronic acid that gets recognized by its series of receptors. And so these are just a few examples of them. You even have, for instance, the use of iron-loaded transferrin as a potential thing that you can coat onto the surface of nanoparticles, because again, recognition by the transferrin receptor, which is actually overexpressed by many cancer cells, given that cancer cells do thrive on the use of iron. All right, so what are the limitations? Well, the thing is that many of these receptors are not unique to cancer cells. And so that being said, because they're not unique to cancer cells, and the reality is that typically you have far more healthy cells than cancer cells, then you have a higher risk of these things being directed towards healthy cells, right? Obviously excluding the EPR effect. Another problem is that a number of these targeting groups are actually important groups in order to enable cell growth. There's a question here. All right, Javier has a question. Make sure that. All right. Is there the possibility for the nanoparticle to lose their nanostructure in the different conditions within the body? So that, yes, Javier, and we're actually going to see a few examples of that in a few slides further on. And actually, that's kind of a desirable thing, because you don't want nanoparticles to be trapped in your body, which is the reason why certain carbon-based nanomaterials are not desirable for the body to the cells. All right, so as I was mentioning, some of these targeting groups are actually essential for cell growth. So in addition to delivering something that might potentially be therapeutic and attack these cells, you also deliver something that will enable their growth. So it's not necessarily the most effective strategy. But the idea is that typically you want to design something that is more of a Trojan horse strategy, where you trick a cell into thinking that it's getting something that it needs, but in fact, you're not. And this strategy would not be an actual true Trojan horse type of strategy. All right, so we're going to just briefly talk about a few types of nanoparticles that exist. and some of their limitations. So I would say to you that some of these limitations, they're looked across at different types of cell lines, because it's a little bit hard, at least in terms of how people have studied these things, haven't studied them not necessarily in the most consistent manner, but we get a perspective of potential toxicity. All right, so with respect to this, we have to talk a little about the cellular transport, although I've alluded this to an extent about how a number of these nanoparticles, especially depending on how you coat them, can be uptaken by cells. And so you have this phagocytosis process, which is basically a type of devouring process in which the cell will engulf the solid particle and internalize it into a compartment known as a phagosome. Then you have the macrophagocytosis, which is basically similar, but here is a regulated form of endocytosis that mediates non-selective uptake of solid molecules, nutrients, and antigens. It is an actin-dependent process that begins from the cell, the surface membrane vessels that produce large endocytotic vacuoles called the macropinosomes. And then we've talked a little bit about the clathrin-dependent or caveolin-dependent endocytosis. The clathrin-dependent one we talked about in the context of the transferrin receptor. So these are receptor-mediated processes, and they undergo stages of progressive acidification, and that acidification process can lead to the release of cargo, but not necessarily. Clathrin helps to protect the endosome from proteolytic digestion to be able to recycle the corresponding receptors. Right, so I mentioned this because these are routes of entry points for a number of nanoparticles. And so this is just a cartoon visual showing you these three main processes: the phagocytosis, the macropinocytosis, and then the more classical endocytotic routes, and how they can ultimately lead to the release of these materials. All right, so with regards to the carbon nanotube, I would say that probably less and less people are relying on these type of materials, just because they can be quite difficult to break down. So these tend to be needle-like fibers used for drug delivery and diagnostic applications, a wide range of length to diameter ratios of up to 10 to the 8th, with different curvatures and morphologies. So we're going to talk a little about their potential risk and fate. I've kind of broke them all down in this context. So structurally similar to asbestos, and so for this reason, researchers have extensively looked into their study of their degradation. So the small compound fullerene, C60, has been reported to be modestly uptaken by marine macrophages, but demonstrate low toxicity. However, single-walled carbon nanotubes, or abbreviated SWNT, are quite cytotoxic. So you have a number of studies, for instance, with the human HEC-293 cells. Those are classically used as assessment of the general toxicity of different materials. Well, show that these nanotubes can inhibit cell growth, can decrease cell adhesion, and can induce apoptosis. And so some of the rationales to how this might be the case is that in the manufacturing process, there may be some amount of trace iron, and that trace iron can become reactive and generate reactive oxygen species in an unregulated manner, or that these materials can aggregate into very large clusters and then have their effects in that way. So a common theme that you will see in this description is that people will try to functionalize the surface in such a way to enhance their delivery capability and also to try to decrease their cytotoxicity. And so here we have an example that this is from a 2013 study. So functionalizing the surfaces of SWNT with organic molecules. So here we're looking at the incorporation of carboxylate or hydroxyl groups that makes the nanotube susceptible to enzymatic degradation by peroxidases. And so that goes into what Javier was talking about, this idea that you want these materials to be cleared from the body, whether your body has a capability of just taking them and clearing them, or has the capacity to break them down in such a way that doesn't lead to the formation of anything that will harm the body and still can basically lead to their being cleared. So this idea is that you're trying to modify the surface in such a way that it can be recognized by certain enzymes that can break them down. And so we're looking at here an example of a molecular modeling study that was done with binding interaction with the eosinophil peroxidase, which is a heme-containing enzyme found in the human immune cells. So they catalyze the formation of hypobromous acid, which is HOBR, and by the use of bromide and hydrogen peroxide. So you're basically trying to take advantage of that enzyme, this particular peroxidase, and to try to break it down. And so this is just a zoom in of these particular two sites, where site one is located near the channel on the enzyme where the substrate bromide binds, and then the nanotube binding of the anionic carboxylate is stabilized by electrostatic interaction. And so what you get as an outcome of this interaction is oxidized polyaromatic molecules, resulting in the formation of carbon dioxide. So to give us a little bit of perspective of the breakdown of this material and what are the actual products that are produced. We also have, in this case, mesoporous nanoparticles. Here we're looking at silicon dioxide. And what I'm showing you here is not actual nanoparticles that are used, but I'm showing you examples from nature. So these are biomineralized silicon dioxide. Nature uses silicon quite a lot, actually. And so diatoms is correct. And even in diatoms, they have the use of titanium dioxide for antimicrobial purposes. But yeah, these are pretty cool. So they have inspired the engineering of mesoporous silicon dioxide nanoparticles. Okay, so these particles have a large number of pores in which small molecules can be absorbed. The outer surface can be readily decorated with functional groups and targeting molecules. And so people really like to use silicon as they can become degraded, but of course there are associated risks with the use of these materials. So studies performed on the human neuroblastoma cells, SK and SH, show that pristine, unmodified MCM41, which is a mesoporous nanoparticle, could be a toxic substance. So different MSNPs, so mesoporous nanoparticles of different sizes, 150 nanometers all the way to 4 micrometers, show toxicity in mesothelial cells at high concentrations, 0.5 milligrams per milliliter over 2 to 3 day exposure. Subcutaneous injection into an animal model demonstrated good biocompatibility and time-dependent release from the body, but intraperitoneal, so that's through the gut, or intravenous delivery to mice resulted in death, possibly due to blood clots, so thrombosis. All right, so you can see some of the effects that we already were talking about before. So surface modifications can attenuate toxicity of these type of materials. Spherical mesoporous nanoparticles functionalized with the fluorescent dye fluorescein was studied to monitor their cellular localization and degradation. All right, so we're basically using this dye to try to understand what happens to this material. What is the biological fate of this material? So these nanoparticles in human umbilical vein endothelial cells, or HUVEC cells, were tracked by transmission electron microscopy, or TEM, and laser scanning confocal microscopy, focusing on the fluorescent properties of the fluorescein. The nanoparticles enter via diffusion and also by receptor-mediated endocytosis and localized to the cytoplasm and lysosomes. And in this process, degradation was observed via both routes of entry. So what is the degradation products? Well, they are the formation of silicon ions in the culture medium, and that basically confirms degradation. So here we have a visual of this process, where you have the entry through an endocytotic process or an entry through a passive diffusion, okay? And then the subsequent breakdown of the materials into these silicon ions. And so that gives you the perspective of how it can be used for delivery of something, but also that the material can be broken down. Okay, so we also have silver nanoparticles. All right, so typically will be of a neutral form, not of a silver plus form, because silver plus, as we talked about when we looked at the antimicrobial property of silver, can be quite cytotoxic. So typically protected by surface conjugation to the thiol-mercaptol-undecanoic acid, or MUA. And so as we know, silver is a soft metal. So you want to use something like a thiol to be able to have a strong interaction with the surface of this. So you use something like MUA, or the polymer polyisobutylene alt maleic anhydride, or abbreviated PMA. So studies on the treatment of NIH3T3 embryonic fibroblasts with 5 nanomolar silver nanoparticles coated with MUA or PMA show localization in the endosome, lysosome compartments, with partial degradation leading to released silver ions. So that actually has the potential to be hazardous, the release of the silver ions. So in a similar capacity, you have gold nanoparticles. So Kaylee may be familiar with gold nanoparticles as well, used for a variety of therapeutic and diagnostic applications. So in a similar capacity as with the silver nanoparticles, the gold nanoparticles, given that the gold is something that's soft metal, surface conjugation to MUA or the polymer PMA. So gold nanoparticles have been used to treat rheumatoid arthritis, as we discussed when we were in that section of the course, since the 1920s, and have been found to be relatively non-toxic to humans. They enter cells via receptor-mediated endocytosis, but they have difficulty leaving the endosome inside the cell. This may be aided by the help of biomolecular coating. These type of particles tend to locate to the trans-Golgi network or to the lysosomes. The particles can be directed to the cytoplasm with a judicious choice of lysine-rich peptide conjugation to disrupt endosome and lysosome interaction. All right, so there was a study that was done to try to understand the fate of these materials. So this was a dual labeling radioactive experiment that was used to track the fate of the 5 nanomolar gold nanoparticles following injection into the blood of rats. So how was this done? Well, the nanoparticles were labeled with 198 gold and also with 111 indium, and that's to track the amphiphilic polymer code of the nanoparticles. So we're tracking the gold and we're trying to track the actual polymer part of the particles. So despite the high colloidal stability of these particles, the polymer coat was found to be partially degraded, and the gold particles to be mostly localized in the liver. So during cell division, some gold nanoparticles are passed to daughter cells, but others are displaced to the extracellular space, indicative of a pathway for particle elimination from the body. So there's some possibilities of the body being able to eliminate it, but also that's a little bit concerning, the possibility that you can transfer it to, the next set of cells. You have a type of nanoparticles, which are the super paramagnetic iron oxide nanoparticles, or also referred to as spions. We're going to talk a little bit about this in our next lecture. But in this context, these can serve as more than just delivery purposes. They can also be used for diagnostic purposes, as they have the capability of serving as magnetic resonance imaging, or MRI, T2 agents. These are typically composed of either magamite, which is gamma iron oxide, or magnetite, another type of iron oxide. And they're typically coated with dextrin, starch, PEG, or other substances. So as you can imagine, they're going to be associated, risk with something that contains iron and that can potentially release iron. So depending on the application, these particles are administered by intravenous, oral, or intranasal routes. Bare particles exhibit some cytotoxicity by facilitating Fenton chemistry because of the iron that composes them. They are uptaken by cells via receptor-mediated endocytosis. They locate to the lysosome, which results in hydrolase degradation of the organic coat, and the low pH can dissolve the iron oxide core. The released iron ions are captured by serum transferrin and eventually can be stored by ferritin or incorporated into hemoglobin and myoglobin. So that's the desirable fate of the iron, that the iron does not lead to just a release that's unregulated, but that it actually can be captured and properly stored. And then the last ones that we're going to talk about are quantum dots. So these are nano-sized particles of semiconductor materials such as lead sulfide, cadmium selenide, and indium arsenide, and other simple binary compounds. When exposed to ambient light, they strongly fluoresce, emitting radiation that can be easily tuned by changing the size and composition of the particle. So the main risk that you're going to have associated with these particular materials is that they can lead to the release of toxic ions. So we're talking about cadmium, lead, and arsenide. So that's the main risk of using these things. And they have the capacity, these particular ions, to bind to proteins and repair their function. And they also can interact with the mitochondria generating reactive oxygen species. So one way that you can try to utilize quantum dots for their delivery purposes, but also trying to maintain their stability, is by coating them with zinc sulfide, which is known to be less biologically active or less toxic. All right. So I had mentioned to you this idea that nanoparticles can come in different sizes, and you're all aware of this. This is just to give us a perspective of how a few different features about the nanoparticles, not just size, but also shape and even charge, can lead to a bit of a difference in terms of potential biodistribution. So if we look at this figure here, and this comes from our textbook, what we see over here is depending on the size, so the diameter, 150 nanometers down to 5 nanometers. If you have the larger size, where you're going to get is higher distribution in the lungs, liver, and spleen. But then if you're looking at the smaller size, right, and that we talked about already, less than 5 nanometers, that can be cleared from the body, and typically going to be cleared from the kidneys. Then if we're looking at the shape, we're talking about the spherical shape, this sort of pellet type shape and this cylinder type shape. Well, even that can have some amount of difference in distribution throughout the body. And so you see that you have, with that more of a pellet type shape, a higher biodistribution in the lungs, liver, and spleen. And then finally, from the perspective all the way to the right, we have the difference in charge, where you have particles, the same in this context, they're all spherical, negative, neutral, and positive charge, and how that can lead to some differences in terms of the biodistribution, where the particles that are positively charged tend to have a higher localization in the lungs, liver, and spleen. So these are factors that can also play a role in terms of how you might want to design things. And so if you look back at the table that I showed you earlier about how to target certain organs, these factors also played a role in tissue targeting. All right, so the last bit of this is I want to talk to you about cytochrome C in terms of this protein, which I've alluded to you before, and how people have tried to use this protein by exploiting its biological activity for a potential anti-cancer drug. And so I'm showing this because of the work that I've done with, in collaboration with Kai Gribenow's lab, in terms of how you try to formulate this protein drug in a way that involves nanoformulation. All right, so a little bit of perspective. And so I'm also bringing this up because I believe it was 2 cycles ago, the Medicine course produced a review article on this protein and looked at many different ways in which people have been trying to develop an approach to deliver this protein for a potential anti-cancer application. So the biological functions of this protein, and you should be aware of this to some extent. So it is a heme-containing protein that plays pivotal roles in cellular life and death processes. And so depending on where it's localized within cells, it will exhibit a certain type of functionality. And also, depending on whether it's phosphorylated or not phosphorylated in terms of structurally, can influence how it functions. So in the respiratory chain of mitochondria, which is located in the peripheral membrane, it serves as an electron transfer protein, contributing to the proliferation of healthy cells. So we're aware of this process, how that is vital to survival. But in the cell cytoplasm, if cytochrome C is released from the mitochondrion, it activates the programmed cell death, apoptosis, and actually is an intrinsic form of apoptosis that can lead to the termination of damaged cells. So it's actually functional for both, for keeping cells alive, but also for signaling the death of a cell. So here's just that perspective. So where you will localize cytochrome C with regards to this mitochondrion, its function within the electron transport, but also its function when it gets released into the cytoplasm. What happens to that protein as it starts to interact with another protein called APAF1, that protein interaction leads to what is known as the apoptosome. That apoptosome formation triggers the caspases to lead to the onset of apoptosis. So the interesting thing about this protein is generally a pretty small protein. It's about 12 kilodalton. So, but it's a very important protein. And so if you look at its evolution, structurally it's characterized by being highly conserved across different species of organisms. Throughout evolution, the sequence of this protein has not undergone considerable mutations, and therefore the domains and residues are crucial for its function and integrity. And so here, what you're seeing is those amino acids that are highlighted in yellow are those amino acids that have been preserved, and you can see them across different organisms, different types of organisms. And so this gives us a perspective of why this is referred to as cytochrome C, because this protein employs the heme C as part of its function. So this is the form of the heme group that's actually associated with the protein itself. So this gives us a perspective that you have the heme group that's located right here. But then the iron center, that iron center has 2 points of contact. You have a histidine that's coordinated here in the active position, and you also have a methionine that's coordinated here. So that's an important interaction with that metal center in order to preserve the functionality of this protein. All right, so let's talk a little bit about the iron center itself. So the heme iron is critical to the protein's activity. The metal coordination enables an iron 3 to 2 redox potential. And so here we have a value of E at a pH of 7 of 250 millivolts versus the standard hydrogen electrode. So this ability to fluctuate between iron 3 to 2 is important in terms of the electron transfer process that it participates in. So it's necessary for the protein to exert its functional role in mitochondrial respiration as an electron carrier between respiratory complexes 3 and 4. But then you can also look at the importance of the metal in terms of its function for apoptosis. So non-iron versions of cytochrome C are widely studied for a structural insight about the protein, and they help to give us a better understanding of the function of the iron. Although I would say that people know very well the importance of the iron in terms of its electron carrier function. But in apoptosis, there's a little bit of a mystery in terms of how exactly it's facilitating this process, how the metal itself does it. But it's known that the metal is important. So the cobalt-3, zinc-2, copper-2 substituted cytochrome C are structurally similar to the iron-bound version. So substituting the iron with these metals doesn't affect them structurally, but there is going to be a difference in terms of the redox activity. So cobalt-3 cytochrome C is redox active and retains some of this iron cytochrome C redox function, whereas the copper-2 and zinc-2 substituted cytochrome C are redox inert. And it's also been observed that the non-iron cytochrome C can induce apoptosis. So these different metal-bound forms of cytochrome C have a capability of apoptosis induction, but with significantly less potency than the iron-bound form. So it's very clear that iron plays a critical role in terms of optimizing the capacity to induce apoptosis, but it's not exactly clear how it's doing that. It's something that requires further study. All right, so this is just another perspective. The only key thing is that depending on how this protein is phosphorylated is how you can see its activity. So here you have under normal conditions where your cells are operating optimally, you have phosphorylated cytochrome C that participate in the electron transfer chain and that enables aerobic respiration. But then when you have stress conditions within a cell, and so what that leads to is damage to the cells. It also can lead to a modification of cytochrome C where it becomes dephosphorylated. And as a consequence of this leads to a permeability of the mitochondria, which releases cytochrome C, and that can result in the formation of the apoptosome and triggering of apoptosis. So depending on phosphorylation, you can have a distinction in terms of the functionality of this protein, but obviously that's coupled with the cellular conditions that's enabling the phosphorylation or dephosphorylation. All right, so to put this in perspective then, we know that cytochrome C, we employ it in our bodies in terms of keeping the cells alive, in terms of possibly triggering apoptosis. Well, people have caught on to that idea of, well, it triggers apoptosis. Well, what about using this protein as a potential therapeutic? And so here's a perspective of different proteins that are being used as potential therapeutic proteins. And so over the years, this number has gone up significantly in the field of FDA-approved recombinant proteins as biopharmaceuticals. And so this is not to say that cytochrome C is actually used in any way as a biopharmaceutical, but there is an interest, and that's why you have a lot of people who've been focusing on cytochrome C as a potential anti-cancer agent. All right, so the fact that cytochrome C is a pro-apoptotic protein for cancer therapy has made people want to try to use it for this application of an anti-cancer agent. But the reality is that protein is poorly permeable. so it can't enter cells. So you have a lot of effort being done to try to engineer cytochrome C in such a way that you can deliver it into cells and try to use it for its capacity to induce apoptosis. So there are important details that one has to consider. For instance, in terms of its size, I already mentioned to you that this is a relatively small protein. So this gives you a dimension of this protein and the factors that you have to think about when you're trying to design around that protein. Right, so it possesses basic surface characteristics. It has a pi, or isoelectric point, of 10.8, owed to its positively charged residues lying on the solvent exposed section. These positive charges are crucial for its activity by interacting with the APOP1 in the cytosol that triggers the apoptosome assembly. So why am I mentioning that to you is because when you want to have that exposed within the cellular environment so that it has the capacity to interact with APOP1 to form the apoptosome. You can't block that. That's a critical site that needs to be available for that interaction. Also, it's known that that there are going to be some cells that won't be sensitive to cytochrome C, because there are some cells that don't have a functioning or let's say a functioning caspase system. So cells that are sensitive to cytochrome C activity possess caspase 3. Another thing to note is that between 40 and 100 femtograms of cytochrome C per cell was found in apoptotic human kidney 293 and MCF7F breast carcinoma cells. So why is this number important? But it gives us a limit of a lower number that you want to be able to effectively deliver into cells. Because if you design something that delivers less than what your cells can release, then that's going to be not something functional. So you need to know what is that lower limit that you want to go above in terms of how much protein needs to be effectively released in order to have any real impact. So this was the work that I did with Kai Gribenau. So this is from his laboratory. And the idea was that we're trying to look at how you can design cytochrome C in such a way that you can enhance its functionality. So the focus of this work, this is from Moraima Morales, this was her thesis project, was that she basically wanted to design a cytochrome C nanoparticle that you can then decorate with different features to help improve the delivery and functionality of the protein itself. Right, so essentially this is the big picture being captured in this visual, where you have the cytochrome C nanoparticle here, and you're coating it with different things that are going to help enable the functionality and the delivery. And so what is being coated with was polymers. Okay, so you have the PLGA, you have the PEG unit, but also what I mentioned to you before, the use of folic acid as a small molecule that can be recognized by receptors. So the idea is that you want to take advantage of this coating to deliver the protein, but then in the actual intracellular environment, this coating will release the protein, and then that protein become functional and be able to trigger apoptosis. But the idea is that, as I mentioned to you, you want to have that protein releasable. You don't want to block it from being active. And so what they did was to use a silyl group so that you can take advantage of the redox environment of the cells to be able to release this protein. Right, so some of this I'm just going to fly through because the main thing is the big picture. We didn't really worry too much about the details of how this was done. But essentially, you want to transition your cytochrome C into a form that's nanoparticle, right? So that's going to be something to think about because not all proteins will be happy going from their stable, soluble form into a form that's nanoparticle. That might actually be irreversible damage to the protein itself. So this step is not a trivial step. You have to design that nanoparticle in such a way that it can become reactivated. So in this case, they use acetonitrile to trigger the formation of the nanoparticle. Then what they did was to use a cross-linker to be able to cross-link the surface of this protein to the agent that contained the folic acid and the PEG and the PLGA. So that's basically what the design. You have the nanoparticle protein, and then you have this whole group here with the PLGA, with the PEG, and then the folic acid. All right, so these are a series of characterizations. I'm, again, not going to focus too much on this, but just to point out some of the features, something as simple as protein NMR to be able to identify all of the different groups that are here that are composing the folic acid, and the polymers themselves. And so one of the things that was useful here was the use of UV-Vis to ensure that you have the formulation that you desire, but also the potential release of the cytochrome C. So here we're looking at the characterization of the folic acid PEG PLGA thiofunctionalized cytochrome C. So this UV-Vis spectra is of a supernatant that was recovered after the coating process. And so the release of the pyridine to thione group produced an absorbance increase at around 280, so here, nanometers, and also at 365 nanometers. So this is just an indication that you have the conjugation of interest that you want, and then also potential to use this as a way to track the release of the protein itself, the cytochrome C. All right, so a few of the characterizations. So here, what we're looking at is sort of the spherical structure of these nanoparticles. And so this is with the full, the nanoparticles and the coating. So you use SCM here to capture that image, but also look at along the way, how you transform the cytochrome C particle and with the coatings, what happens to the zeta potential. So you can see that with every modification, there is a change in the zeta potential. So when you're going from the just the cytochrome C nanoparticle, we're at a zeta potential of about 15, all the way up to when you have the full conjugated functionalized cytochrome C up to about a value of 50. So there is a significant change in terms of the zeta potential of this actual structure. All right. So here we're looking at then the functionality of this. So as I was mentioning to you, you want to be able to release this the cytochrome C from the way you've designed this. So the idea is to probe how the redox environment of the cells can influence this. And so here we're looking at the use of glutathione as sort of a model of the redox environment of the cell. So we're not looking at this within the cellular environment. We're simply looking at how the different concentrations of glutathione can potentially lead to the release of cytochrome C. So we're tracking release of cytochrome C in the presence of no glutathione, 1 micromolar glutathione, and 10 millimolar glutathione. So in our blood, we have about micromolar amounts of glutathione, and in our cellular environments, we have 10 millimolar, and actually even higher in a cancer cell. And so we can see we have low percentage of cytochrome C released in the low amount of glutathione, but a significant amount of cytochrome C released in the presence of 10 millimolar glutathione. So that indicates that the redox environment within the cellular environment should be sufficient to release the coating from the cytochrome C. But that doesn't mean that that protein is functional. That only means that you can release the protein. So what we're looking at to the right is the capacity of the protein to be able to trigger activation of the caspases. Because remember, cytochrome C uses the caspases to induce apoptosis. So what we're looking at here is the activation of the caspases as a consequence of the cytochrome C. So this is a non-cell assay. You can remember that in order to assess activity, and if you want to work with cytochrome C, the native form is not cell permeable. So this is, you're taking the enzyme and you're seeing if it gets activated in the presence of the enzyme and also in the presence of the nanoformulated cytochrome C. So what we're looking at here is this is done by a kit that you are able to look at the absorbance characteristic of activation of caspase 9 and characteristic of the activation of caspase 3. And so what we're seeing here in the black, this is due to the native cytochrome C. And actually, I don't recall they use horse or human. They're pretty compatible. And so I think the horse one is much more cheaper than the human one. So you can see that the native cytochrome C is quite active. And then you have the cytochrome C that's been functionalized in this nanoparticle formulation with the 10 millimolar glutathione. And you see that it has similar, although not identical, capacity to activate both of these enzymes. Okay, so there's difference, but you still see that it has activity. So the release cytochrome C is active. It has the capability of triggering the caspase. And then you have a control. And so this control here is the same formulation, but now using cytochrome C, you're using I think it's albumin as a protein that's been made into a nanoparticle. And as you can imagine, you don't really get much activation because that's not something that's known to have that capacity to activate these caspases. Same thing here. Excuse me. All right, so here is a study that was done to try to image a cellular uptake of these materials. And so you're using DAPI, which can coat the plasma membrane, and the propydium iodine, which will coat the nucleus. And so what we're tracking here is the presence of the actual protein as it can reach the intracellular environment. So is this actually burned inside the cell? So what are we looking at here is untreated cells. So no cytochrome C, no nanoparticle formulation cytochrome C. We have here just the polymer with the folic acid. Okay. Then we have over here the control with the albumin nanoparticle that's coated with the polymer, and then the cytochrome C that's coated with the polymer. Okay, so this is what we're tracking, and then we can see that we can label all this the plasma membranes with DAPI. So in every single case, we have the plasma membrane being labeled, but then only the one that has a cytochrome C do we see that we're actually able to label the propydium iodine, which is where we're looking at the localization of this. So study of DAPI and propydium iodine co-localization for detection of apoptotic cells, selective induction of apoptosis observed in HeLa cells. So these are the type of cells that we use, forgot to mention that, incubated with the nanoparticle formulation of cytochrome C. No cellular apoptosis observed in untreated HeLa cells or when incubated with the control. So only apoptosis observed in the presence of cytochrome C. And how do we know this? Because with the capacity to be able to label the nucleus is an indicator that we're going through this process of apoptosis. All right. So what we're seeing here then is the effect of the formulation on cell viability. So previous slide was, is it even entering to the cell and is it inducing apoptosis? But here we get a perspective of the effect in terms of the viability of the cells. And so what we're looking at is percent viability compared to untreated cells. And then we're looking at different concentrations of cytochrome C in mg per milliliter. And so this is going to be with folate. Okay, so the formulation nanoparticle of cytochrome C and without folate. So the idea is that we want to see if there's a difference, whether or not you have the folate. Is the folate doing anything? And so we're treating them in a cell line that has a folate receptor. So the idea was that you want to take advantage of that receptor to see if there's any selectivity. And what's observed is that you have a much lower cell viability in the presence of the nanoformulated cytochrome C when folic acid is available. You can see this dose response, and you can see how you get to the level of about 20% viable at the highest concentration of the cytochrome C. You do get somewhat of a dose response without the folic acid, but it's much less. So you get about, 80% at the highest concentration. This is indicative of the folic acid enabling possibly a higher uptake. And possibly that higher uptake is the reason why you have a lower cell viability there. And so the last image C is basically a confocal fluorescence study, images of HeLa cells after incubation for 6 hours with FM464. Basically, that helps to label endosomes, and then FITC label cytochrome C. So we're basically looking at the endosomes, because presumably this is going through the folate endocytotic process through the receptor. And we're looking at FITC. FITC is what's been used to label the protein. And we see as a co-localization of the endosome and also the FITC to some extent, the co-localization of this as a consequence of that being brought in through endocytosis into the cellular environment. And so this one is basically another perspective that this is a little bit harder to see. But the idea of this is that you want to test selectivity against the HeLa cells, which the HeLa cells have the folate receptor, A549, which is a long cell line, which does not have an overexpression of the folate receptor, so it's deficient in that receptor. and NIH3T3, which is a non-cancer cell line, which relative to the HeLa cells has a lower expression of the folate receptor. So basically, we're looking at a 6-hour incubation of these cells with the nanoparticle formulated cytochrome C, and we're just tracking the uptake in the cells, and only the one that has the actual, the folate nanoparticle cytochrome C is observed in the HeLa cells and not in the other two cell lines, which indicates that there is some amount of selectivity for the folate receptor containing cell lines and not the other two. That doesn't mean that they don't have the capacity to take up some of it, but from this perspective, there's more selectivity for the HeLa cells. Okay, and this is another perspective of this same study. And what we're seeing is that over time, how much of that fluorescent intensity due to the cytochrome C, we see that we get a much elevated concentration of the cytochrome C within the HeLa cells relative to the A549 and relative to the NH3T3. All right, so here's another study that was done, and this is a cellular uptake visualized ex vivo. So a study was done with an animal model, and we're looking at brain slices to see if this particular formulation of cytochrome C is able to get into the tissue itself. So it's a little bit hard to see, a little bit blurry, the image even on my screen. But what we're looking at here are brain slices from the glioma GL261 tumor-bearing mouse. Showed more intense fluorescent signals of FITC labeled cytochrome C-based nanoparticles in the tumor cells in comparison with the healthy brain tissue. So we're looking at brain tissue and healthy brain tissue. And we see in the area where there's a tumor is more lighting up due to the FITC from the cytochrome C. And there's another perspective of how much fluorescent intensity is from the tissue that's associated with the tumor as opposed to the non-tumor tissue within the brain. All right. And the last bit of information is this in vivo application where we're looking at the effect that the actual cytochrome C formulated in this manner, how it may potentially be useful. So I would say that this is probably not the most impressive result where we're looking at the in this study where you have the tumor size in these C57BL6 mice that were implanted with these GL261 glioma tumor. And we're just looking at what happens to the size of the tumor over this time frame with different, with this direct infusion of the compound, 1 microliter per hour, 100 milligrams per milliliter over 3 days. And so we're looking at this, the image in terms of the tumor size after 7 days. And so looking at this, we see that compared to the tumor size without treatment, this is just PBS control, we see some amount of reduction of the actual tumor size relative to that. But I would say that this is a little bit hard to tell because there's not a direct side-by-side control. What I mean by that is that we're not looking at the percent difference from points when the tumors were present to where they are at this point. So we don't know how much of a percent difference. So maybe a little bit misleading and also not that impressive of a drop. But you can see that there is some amount of response with respect to the use of this nanoformulated cytochrome C. All right. So before we head out, do you have any questions? Any doubts?
Los materiales que se presentaron también se utilizan para mejorar las propiedades mecánicas de la movilidad. Yo me pregunto si esos riesgos hay la vida también pueden ser utilizados.
See, so some of those cell lines that were studied were actually skin cells. And so there are associated risks, yeah, as you would imagine. See, see it's a lot of treatments that have to do with like, like exposure to light, you want to be careful with. So yeah, these are things that you have to think about.
Aunque no lo lleve puesto, porque si trabaja con él a diario cerca de algo que está, se va a poner.
Other doubts, questions? All right, then, I look forward to reading your papers.
presencial.